This is the 35th video presentation for the CASE Plant Science course. As you view this video, make notes of important information. Pause, repeat, and resume the video whenever necessary. Principles of Why It Works, Lesson 7.4, Plant Multiplication. You have completed a variety of asexual propagation techniques through this lesson. Asexual reproduction is commonly associated with plant reproduction, although some simple animal species can reproduce asexually as well. Asexual propagation is an efficient and economical option for many species of plants. Some sterile cultivars of plants can only be propagated asexually. It is important to understand how asexual propagation is used in the plant industry. This presentation provides the steps and processes related to cloning plants from diploid tissues. A review of the asexual propagation methods studied through the lesson. The products of all asexual propagation methods are genetic copies of the parent plant or clones. How does asexual propagation actually occur in plants? What processes take place at the cellular level? Pause the video to respond to the question in your notes. Resume the video when you are ready to move on. Two ways that plant growth occurs are by cell multiplication and cell elongation. Cell multiplication is significant in asexual propagation, especially micropropagation, since the new plantlets are comprised entirely of newly formed cells. In order for healthy plant development, the three basic building blocks, water, nutrients, and hormones, are required. Plant development requires optimum conditions. Cell multiplication is driven by the presence of nutrients. Hormones aid in the formation of cells that contribute to specialized functions like roots, vascular, and meristematic tissues. Water is critical for a plant to maintain cell function. As you discovered with cuttings, water needs to be managed to prevent cells from drying out and dying. New cells are especially susceptible to drying out because specialized plant structures, such as the epidermis, are not formed yet. Pause the video and respond to the practical thinking questions in your notes. Resume the video when you have completed your responses. Conventional asexual propagation methods use mature plant material that has stored nutrients and hormones to support the needs of the developing cells. Because some methods use such a small portion of the plant, though, there may not be enough nutrients present. When dormant plant material is used in propagation, hormones may not be present, or may be present at very low levels. In this situation, a rooting powder containing hormone can be applied to ensure adequate rooting. The sources of nutrients can be attributed to different factors based on the method of propagation used. Pause the video while you respond to the questions in your notes. Resume the video to clarify your answers. When cuttings develop, the plant cells are nourished by the stored food reserves in the stock material. During budding, division, grafting, and layering, the plant cells are nourished by the functioning parent. To reinforce the role that hormones play on plant growth and propagation, you need to remember that the anatomical structures a plant depends on for survival rely on hormones for formation. Hormones affect the rate of growth and development as well as the formation of specialized cells for tissues. As with nutrients, plants have different requirements for hormones based on the amount of plant tissue used in the propagation process. Pause the video while you consider these questions and respond in your notes. Resume the video to clarify your answers. When cuttings develop, hormones are usually added to the cut to encourage rapid root growth. During budding, division, grafting, and layering, all of the hormones needed are transferred from the parent plant. Another method of asexual propagation is micropropagation. Micropropagation uses a minute portion of plant tissue, typically meristematic tissue, that most likely does not contain enough nutrients or hormones to support cell multiplication and formation. During micropropagation, Specialized nutrient auger provides water and nutrients to encourage cell multiplication. Once a callous, unorganized group of cells is formed, an auger containing nutrients and auxins, which are plant hormones, is used to force the tissue to develop into specialized structures, including roots and meristematic tissue. Because of the intensive process and the prolonged time required to successfully propagate plants through tissue culture, you will not attempt the process during this course. The CASE Animal and Plant Biotechnology course offers the opportunity to perform tissue cultures. As a review, mark or highlight 3 key points in your notes that are important to remember from this presentation. List 2 ideas or concepts that relate to previous knowledge. List any questions you have about this topic. Discuss these questions with your instructor. Keep any notes you have from this presentation organized and available for use throughout the course. As the culmination for all of the plant reproduction lessons during Unit 7, you will develop a reproduction demonstration for one species in your grower's handbook. Include this demonstration in your final portfolio. Your teacher will share additional instructions for this demonstration.